Good morning. It's Friday. It's 8:30. I would like to welcome all of you to celebrate vision research. It's again one of the most beautiful days to do research. And today we will be inspired by a wonderful speaker. So with that, I would like to pass to Zach to introduce him.
Good morning, everyone, and welcome to this week's Friday seminar hosted by the Center for Translational Vision Research at UCI. My name is Zach Engfer, a PhD student in the Palczewski Lab, and it's my pleasure to introduce our speaker today, Dr. Jianhai Du. Dr. Du received his master's in physiology from Lanzhou University and his PhD in pathophysiology at Peking University before going on to do 2 postdoctoral positions. His first position was under the mentorship of Dr. Scarlett Jianhai at the Medical College of Wisconsin, and his second postdoctoral position was under the mentorship of Dr. James B. Hurley at the University of Washington. During these postdoctoral training periods, Dr. Du conducted research on signal transduction pathways in cardiac cell types and characterizing enzymes responsible for maintaining metabolic homeostasis in the retina. Following his postdoctoral training periods, Dr. Du went on to serve as an instructor and research assistant professor at the University of Washington before joining West Virginia University as an assistant professor. Dr. Du currently serves as an associate professor in the Department of Ophthalmology and Visual Sciences at West Virginia University and has published high-quality journal articles on the biochemistry, metabolomics, and physiology of retinal cell types. Additionally, Dr. Du has been awarded several R01 grants to fund his research on proline and pyruvate metabolism in the retina, and has been a mentor to many graduate and undergraduate students since joining his appointment at West Virginia University. Today, he will give a talk titled Cell-Specific Metabolism in the Retina. I personally am very curious about the differences in cellular and metabolic resilience between neuronal and non-neuronal cell types within the retina, and how that influences progression of retinopathies. I'm looking forward to hearing more about how specific metabolites help to maintain cellular homeostasis in different retinal cell types from Dr. Du. Please join me in welcoming Dr. Du.
Good morning. Thank you for introduction, Zach, and thank you for the invitation. It's such a pleasure and honor to present my work in this seminar series. So let me share the slide first.
We see the present.
So everyone can see well?
Yes.
Yes.
So today I want to share some of my work on cell-specific metabolism in the retina. So we all like food. I know you guys are like breakfast now, and some of them are vegetarian, some of them are meat lovers, but we can choose food because there's so many kind of foods we can choose. And the choice of food can shape our health. But at cellular levels, the cells have abundant food to choose as well. We are curious to ask a question whether cells have the choice of their own food. If you look at the gene expressions of the single cell sequence databases, the metabolic gene expression are really specific in different cells. It looks like the cells can have their choice of the food. For example, in RP cells, the lactate transporters, or even the ketone body genes, that is a key gene in ketogenesis, they are mostly in RP cells. If you look at the sugar transporters, they can transport glucose, fructose, and they're mostly in neurons, specifically in photoreceptors. And if you look at the glycolysis genes, and they're really high in the photoreceptors, especially in cone cells. And some of the glycosylation genes, they're almost specifically in the cone cells. So our lab is interested in how these nutrients in different retinal cells, they are transported and utilized and coordinated and recycled, and how the impacts on retinal metabolism, visual function, and retinal degeneration. As Zach said, today I talk about the neuronal cells and the non-neuronal cells, RPs, and compare their specific metabolism. Because we cannot separate the photoreceptors from the retina. So here is mostly we're using neural retina as a model to study. And we try some ways to understand the photoreceptor metabolism by these neural retinas. So the neural retina, they are really special because they have the capability to sense the light. And they require rhodopsin, cyclic GMP, and the glutamate as the neurotransmitters. And it has known for many years, the phototransduction process, especially photoreceptors, are really expensive. So how many ATP they can use is surprisingly high. So each second, one rod of photoreceptors can use about 100 million ATP per second. So that is about 10 to 20 times higher than average mammalian cells. And in the cone cells, they can be even higher. It's about 1.5 times higher than the rod photoreceptors. So how our body powers this process of phototransduction? And glucose is major fuel for this process because glucose is important for glycolysis, mitochondrial metabolism. For some enzymes like rhodopsin and many other enzymes, they need to put a sugar coated this protein called glycosylations. Also for ATP and the second messenger cyclic GMP, the synthesize require glucose, also require some amino acids. So that is why the mutation of the genes in glycolysis or in nucleotide synthesis can cause photoreceptor degeneration. Retina also has really higher biosynthesis. It is known that the photoreceptor outer segments has continuously shed and phagocytosed by RP cells. About 7 to 10 days, all the outer segments are new. So basically, every day when we wake up, we have almost brand new eye. If you look at the photoreceptor outer segments, almost 50% are lipids, and the rest of the proteins. If you look at lipids, and 90% are glycophospholipids and some cholesterols. And the lipids have different species like PC, PE, the major part. And there's a small amount of PS, PI. And most of the carbons coming from glucose. And some are from fatty acids. We know for this PE and PC and PE, almost half of the side chains are DHA. And for the desaturation process, they also need glucose to provide the NADH. If you look at the proteins, almost 90% are rhodopsins. And they're also transducing arrestin and other important proteins for phototransduction. But the distribution of the amino acids, we know there's 20 amino acids to make proteins, but they are not distributed equal. The primarily alanine and also leucine, isoleucine, and the valine, those are branching amino acids. They are glutamate, glycine, lysine, and prolines. So glucose is really important in retinal metabolism. But interestingly, glucose metabolism is really special in the retina. Otto Warburg has found it in 1920s in tumor cells as well as retina. They have really special metabolism called Otto Warburg effect. So in the Warburg effect, glucose is will be metabolized into lactate instead of going to pyruvate to the TCA cycle. So when I was at Jim's lab, we compared retina and with other brain slices. So retina indeed has really a higher rate of glycolysis. Almost 90% of the glucose they're going to lactate. So what's the advantage of Warburg effect? Aerobic glycolysis. From tumor metabolism, in the past 10 years, people start to understand this Warburg effect will give advantage for anabolism or synthesis. It's important for lipid synthesis, nucleotide synthesis, and amino acid synthesis. Also, there's recent publications showing in the photoreceptors, this Warburg effect is also important for outer segment synthesis. Retina has really small amount of pyruvate going to mitochondria. So when I started a lab, I asked a question, how important this pyruvate get into the mitochondria? So we generate knockout mice to remove key transporters that transport pyruvate into the mitochondria, called mitochondrial pyruvate carrier. So when this carrier is locked out, the pyruvate accumulated because they cannot get in mitochondria. But also we see higher aspartate and a very low glutamate, glutamine, and glutathione. And recently, we collaborated with Dr. Brian Jones to see the distribution of these metabolites. And similarly, we see glutamine, glutamate, and glutathione that decrease in the photoreceptors and the mural glial cells. So this data is showing that glucose metabolism is really important in the neurons, especially in photoreceptors. And the small amount of pyruvate in the mitochondria also critical for the neurotransmitter synthesis. So what about the glucose metabolism in vivo? So we collaborate with Dr. Douglas Dean. He has a very nice pig models. So this pig looks small, but actually it's not very small. They put catheters in all the way to the ciliary arteries and also connect the metabolites from vortex vein. So all the autoretina, most of photoreceptors, their nutrients coming from the ciliary artery and drained by 4 vortex vein. So we collect the blood in the early morning and afternoon and look at glucose and lactate uptake. So it's interesting, in the morning, there's more glucose is uptake than the afternoon. And the lactate is more generated in the afternoon. So this data suggests that glucose is important for the outer segment synthesis. So we know a lot about glucose metabolism. So we are interested what about other nutrients that can require it? And also we know the cone photoreceptor is really important in the retina. So we're interested what is the metabolism or metabolic needs between the rods and the cones. We take first we take a buffet approach. So we give rich nutrients. So in the retina, then after 24 or 48 hours of incubation, we look at what nutrients are taken. So then we know what nutrients are used. So first we collaborate with Dr. Wei Liu from Albert Einstein University. He has developed very nice model for retinal organoids. That is cone-rich retinal organoids when they grow for 296 days. And about 56 days, they have organoids but no specific retinal markers. We compare about what kind of nutrients the uptake from this rich media. If you look at the 56 days and 296 days, that is mature retinal organoids. It's really stand out that the mature retinal organoids, they use more aspartate and glutamate. So what about the retina in vivo? So we collaborate with Dr. Anna Swarup group. They developed NIL lockouts. So in regular photoreceptors, almost 97 of the photoreceptors are rod photoreceptors. But when NRL is locked out, in these mice, most of the rod photoreceptors are converted to cone-like photoreceptors. So we do the same experiment, incubate this retina for 24 hours and collect the media to see what nutrient they use. They can use glucose and many other nutrients, but that is really stand out, is very similar to retinal organoids. Almost 10%, almost tenfold of the aspartate and glutamate are decreased. If you look at the NIL lockouts compared to the wild type, the rod dominant cells, they use more pyruvate and cysteine. So why they use too much pyruvate? We look at the enzyme, the gene expression of these NI lockouts. They express more genes related to pyruvate metabolism. Maybe because corn need more energy, the glucose not enough. They need a pyruvate. Cysteine is a precursor for taurine, hypertaurine, cysteines. So they may need more for the corn metabolism. So what about the human retina? So we collaborate with Dr. Mark Gillies. They have the largest eye institute in Australia. They can collect fresh postmortem tissues and make punches of macular and peripheral regions. We know the cone photoreceptors mostly reached in the macular regions. So we do the same experiments and compare the uptake of nutrients in the macular region compared to peripheral regions. And similarly, we can see they use lots of aspartate and glutamate, almost like 20 times or 20-fold decrease of aspartate glutamate. When we compare macular and peripheral regions, similarly, we see in the macular region, they use more glucose. Also, they use more pyruvate. This is very similar to our observation in the NIL lockout mice. And also, in the macular regions, the gene related to pyruvate metabolism is higher in the macular regions. So why they use so much glutamate and aspartate? We think they can be important nitrogen sources. In the retina or photoreceptors, they need a large amount of ATP, and they need lots of cyclo-GMP, glutamate and neurotransmitters. They need lipids for other segments and also need a lot of proteins for other segment turnovers. From what we learn, glucose is primary sources, and we know retina can use ketone bodies. Retina also can use fatty acids, but all of this can only provide carbons or nitrogens. The carbons, they cannot provide nitrogens. But if you look at these molecules, they are nitrogen rich. We think the aspartate and the glutamate may potentially provide nitrogens to synthesize these molecules. So in short summary of the neuroretinal part, the glucose is the primary fuel for bioenergetics and biosynthesis, and the retina can utilize aspartate and glutamate. And the cone-dominant retinas need more pyruvate. So now I want to talk a little bit about RP metabolism. It's long neuronal cells. RP is monolayer cells that is lying between photoreceptors and choroid blood. It has many important functions like phagocytosis, transports vitamin A, and secrete some growth factors, and also important for transport nutrients and metabolites. RP is very important because when RP dysfunction, especially when its metabolism not function well, they can cause photoreceptor degeneration, and it's closely associated with disease like age-related macular degeneration. So why RP metabolism, they are not a function very well, they can cause photoreceptor degeneration. So what is specific features of RP metabolism? If you look at RP, they are really different from photoreceptors. RP, they live in very specific regions. They have very good access to many nutrients. From the blood side, they can access fatty acids, the amino acids coming from the blood sites, and also they have glucose. Every day, they also can accept the nutrients from the photoreceptor side. We know that 11-cis retinol, they can recycle in RP, and the lactate, they're coming from the glucose because the high Warburg effect in photoreceptors. There's also a lot of proteins and lipids from other segments, so they can be further degraded, and this degradation has to be further processed, and finally, they will become NADH and FADH. They have to be recycled, and the major center for recycling NADH or FADH are RP mitochondria. or small amount by peroxisome. So that is, we think the RP mitochondrial is super important, or mitochondrial metabolism is really important for RP as well as for photoreceptors. Because when they're not manufactured, degrade this NADH very well, they can potentially cause accumulation of lipids or proteins in AMD. So because they have higher reductive environment, is higher NADH, we found in RP, they have really special metabolism called reductive carboxylations. Also we call reverse TCA cycle. Most TCA cycle is clockwise, but in RP, they have this reversed TCA cycle to counteract this higher reductive environment. They can move the NADH from the mitochondrial into cytosol and convert to NADPH. In this way, they can combat this oxidative stress. So how RP use different nutrients? We use the same strategy. Actually, we use the same eye from those we incubate to look at the neuroretinal metabolism. We look at we take the RP and the choroids and look at their uptake of nutrients. If you look at the nutrients uptake, they're quite different from the neuroretinal. They do not use too much aspartate and glutamate, but they use succinate, as Dr. Jim Hurley reported a couple of years ago. And they can use proline, lysine, the leucine, and the valine. And also they can release many nutrients. They can release mitochondrial intermediates. They can release ketone bodies, and they can release cholesterols. In the macular region, they can use more glucose. They can use more nicotinamide that can use as precursor for NAD synthesis. They can also use more erythritol that sometimes we add in the coffee. Actually, they can be metabolized as well. So to comprehensively study how RP use different nutrients, we come to the primary RP culture. So we collaborate with Dr. Jennifer Chow from University of Washington. It's difficult to study the nutrients uptake in the RP culture because we do not know which media is the best, because the nutrient consumption, it depends on availability. For example, now you have donut, you have to eat a donut. If you have something else, maybe you choose something else. So we choose the three that is most popular media used by different labs. The first one is MEN-based culture media. And this media is really popular. It's used by NIH group, originally developed by Sheldon Miller. And the DMF12 is used by many labs, including like David Williams and a couple of other labs. Ex vivo 10 is a commercial media. We also include HPLM, or human plasma-like media. Low one uses to culture RP. This media is developed by the cancer research group. In cancer field, they find many of the metabolic studies are a little artificial because the media is artificial. So they develop this human plasma-like media. And we add this media with serum or with B27. And after we grow this media for 4 or 6 weeks, all the RP cells can have mature RP markers with good TERs. And we interestingly look at the nutrient consumptions. Interestingly, all these 6 medias, they can, in these 6 media, they can use proline, isoleucine, and leucine. It's very important is for the proline, almost all of proline are consumed. It's about 400 micromolars. In the isoleucine and leucine, almost half of them are used up. If you look at many other amino acids, they are quite different by different media. But aniline and glutamate is really different. For all the media, they can export it, aniline and glutamate in the media. For glutamine, it depends. In the Sheldon Miller media and the media 2, they can consume glutamine, but for others, they can produce glutamate. Then we come back to prolines, because we thought proline can be important for the RP metabolism. So we are interested in how they are important for the RP differentiation. When RPE grow in vitro, they will become pigmented at different weeks. Week 1, week 2, you see the week 4, they become more pigmented. And we look at all the amino acids in the media. And only proline, they are time-dependently consumed by these cells. So when the cells gonna mature, they start to use more prolines. We take another strategy by put more different cell numbers in the when we grow these cells, because when we grow more cells, these cells more mature faster. And we can see is only proline compared to many other amino acids that dose dependently or like cell number dependently uptake. So this also suggests that proline is related to the RP differentiations. So the proline metabolism seems really associated with RPE. In the mature human fetal RPs and iPS RP cells, they use prolines. But in ARP19 cells, they do not use too much proline at all. Except when we add some nicotinamides. There's a group from David Williams groups, they add nicotinamides and make ARP19 cells a little, have some more markers like mature RP cells. And they start to use some prolines. But for the cell line like human RP1 or HEC19 293 cells as a control, they do not use proline. Actually, they can produce proline. So what about in vivo? So we look at artery and vein from these peaks. So similarly, we see proline was consumed in compared to the arteries. But actually, the glutamine can be generated. And this data further suggests that proline may be important for the RP metabolism. Then we also look at the gene expressions. When we look at the human retina and RPE gene expressions, very interestingly, the proline related gene are highly in the RPE. But the aspartate and the glutamate metabolism are highly in the photoreceptors. So we think RPE may catabolize prolines to support the neuroretina. To study this, we isolate the mouse retina and RP choroids and incubate with 15 nitrogen labeled proline, so we can track where the proline is going. We incubate tissue at different time and analyze by mass spectrometry. Based on the literature, proline can potentially contribute it and label glutamate, glutamine, many other amino acids. And indeed, in the RP choroids, that is in the black lines, we can see this 15 nitrogen can convert it into glutamate, aspartate, glutamine, and many other amino acids. But the red lines represent the retina. In the retina, we did not see any incorporation of the 15 nitrogen from proline, suggesting that retina do not take proline for catabolism to make these amino acids. And RPE can use it to make these amino acids. So then we ask the question, is possible the RPE make these amino acids with proline, then exported these amino acids to the retina, and the retina uses amino acids. To do these experiments, we culture mouse retina and culture human RPE. Then we put RPE and the retina, culture them together using the same strategy, put 15 prolines. We look at how much the 15 unlabeled proline is labeled. The fraction is how much percentage. For example, the 90% are prolines, so it means 90% are labeled prolines. Also look at how much concentration they can convert it to these amino acids. So the red represents the retina only, and the yellow line is represent the retina with co-culture. The blue represents RP or RP with co-culture. If you look at the red lines, except proline, you cannot see any other amino acid got labeled. But if you look at this yellow line, you see they start to pick up these amino acids from prolines in the co-culture. It's more obvious if you look at how much photo change of labeled amino acids. Whenever they got co-cultured, the retina start to take these amino acids, and these amino acids can only be made by the RP cells. Except the GABA, a little bit different. The GABA probably coming from the retina and can feed the RP cells. Similarly, if you look at the culture media, the media compared to the RP only, the co-culture media, this labeled amino acid become less, suggesting that these amino acids that are made by RP, they are indeed used by the retina. So here is a short summary of this experiment. If we use retina only, we give them labeled proline. There's almost nothing or very slow of this 15 proline going to amino acids. But if we grow in RP, the many of the proline are converted to amino acids. But when we do co-culture, the amino acids that made from proline not only going to the RP, but also can be used by the neuroretina. So what about this in vivo? To study this in vivo, we fast mice for 18 hours, then inject with 15 prolines, then we connect at a different time. Look at the prolines, because just like one shot. After about 5 minutes, the fraction start to drop. This is the black line is RP, the red line is the retina. This suggests that the proline is quickly metabolized. And you can see the proline can convert it into glutamine, oxoproline, aspartate, glutamate, and many other amino acids. Interestingly, they can label both. It's different from the in vitro. If in vitro we give retina only, they cannot convert any proline into amino acids. But in vivo they can. But it's interesting in vivo, you can see the in the RP, they start to increase to other amino acids, but then the drop at 15 minutes. But for the retina, at 5 minutes, you almost do not see any incorporation except glutamine, the small amount of incorporation. By 5 minutes, pretty slow. But then in the RP, they are pretty high, and at 15 minutes, they start to drop. But in the retina, even at 30 minutes, they still continue growing. So this suggests that RP may be a manufacture center, and retina is consumers. So it looks like when we place order in Amazon, and they take some to manufacture, then to ship it to the users. So that is why in the photoreceptors or retina, they keep growing. To further confirm this, we try to knock out one of the key genes called proline dehydrogenase in the proline catabolism. So proline can catabolize in P5C, glutamate, and glutamine by these key enzymes. By doing Western blot, we find these key enzymes is primarily expressed in the RP choroids, not too much in the retina. It's about 15 times higher in the RP than the retina. And we generate this PLDH lockout mice and validate it. They are really lockouts in the RP. And we do the same experiments. When PLDH is lockout, we see very little proline converted into glutamine, aspartate, glutamate, oxoproline, and many other amino acids we did not show here. So this result further suggests that RP is the site that proline got metabolized to make amino acids, and these amino acids can come into the neuroretina. So why RP use so much proline? We then ask a question, use some mechanism proline can transport. So we look at the literature to look at what are the key genes in the RPE. There's one studies, they find 154 genes. They are specific in the RPE. There's one transporter called SLC6A20, actually is one of them. We look at another data set, they find in human RPEs, there are 171 genes, that is signature RPE genes. And in the mouse, there's 64 genes. When they overlap together, only 22 genes, including RP65 and several key RPE genes. And one of them is this proline transporter called SLC6A20. We also look at the expression of all the potential proline transporters in primary cultured RP cells. We know when the culture for the maturation, they start to use prolines. So look at which transporter can increase. There's only one transporter, that is SL6A20, is increased over time with culture. Whenever they have more passages, the cells lose their RPE markers, and this transporter not increased anymore. Also, recent study showing that this SLC6A20 can be an important new loci for macular degeneration. And they also find in the McTail disease is another macular degeneration. The SL6A20 is a new loci. So all these studies support the proline metabolism can be important in retinal disease. And we validate this. Will this SLC620 express? And we're using antibodies in human tissues and in the mouse tissues. We find they are mostly expressed in the RPE, but not in the retina. Also, recently we generate RP-specific flux lockout of SL6A20. When we lock out, we did not see this. We still are in the processing of categorizing the mice. And we hopefully can show this data later. So in summary, we find in the RP cells, they can use a lot of prolines, and the proline can, through PRODH, to generate many amino acids. So we find about 13 amino acids they can generate by the prolines from the RPE. And these amino acids can transport it to the photoreceptors, use it as neurotransmitters, and carbon can be used for energy metabolism. They can be important building blocks for the outer segment synthesis. Finally, I want to show some new data. We're using another strategy to screening the nutrient utilizations. Instead of the buffet method, we try the a la carte manual method, just give one nutrient at one time. So we give one nutrient in each 96 wells and give very limited nutrient media in human RP cells. And we screened 91 carbon sources and then 92 nitrogen sources to see what capability for these RP cells can use. Then we use redox diodes to test what kind of nutrients they can be used. One of the redox diodes got a light up, that means this nutrient can use in the mitochondria to generate NADH. So first we test the mature human RP cells. In the mature human RP cells, they can use many nutrients. For carbon sources, they can use about 23 carbon sources. In the nitrogen sources, they can use about 24 nitrogen sources. So interestingly, they can use sugars, not just glucose. They can use galactose, mannose, glucose 6-phosphate, that's intermediates for glycolysis. They also can use nucleotides. Nucleotides, they can have the ribose in the nucleotides. They can use for glycolysis. They can also use pyruvate and lactate. They can use ketone bodies, fatty acids, succinate, and propionate that can also go into succinate. For the nitrogen sources, they can use free amino acids as well as many of the dipeptides. And from amino acids, they can use proline as well, and use ornithine. So this pathway is really active in the RPE metabolism. Further support the proline metabolism can be important for the RPE. So what about the dedifferentiated cells? So in the dedifferentiated cells, they use much less nutrient, only about 15 to 30% of the carbon sources and of nitrogen sources. So whenever the RPE, they are mature, they're much more flexible. They can use many nutrients. It's like a young, healthy man. They can eat almost all the foods. But whenever they are got sick, they're only specific for sugar. And all specific, if you look at these amino acids, almost all glutamine or glutamine-related peptides. So look at the gene expression of this fibroblast-like dedifferent RPE cells. In these cells, they are highly expressed of genes for glycoproteins. They can build ECMs, and they have actually higher gene expression for making proline. But for the proline degradation process, all decreased. And genes in mitochondrial metabolism are decreased, but in glycolysis, they are increased. And the transporters for lactate, pyruvate, hidden bodies, all decreased. And that may explain they cannot use lactate and pyruvate or hidden bodies in these fibroblast-like cells. So finally, we want to test whether we can apply this platform to test patient cells. We collaborate with Dr. Jennifer Chow. She's a physician scientist. She has patients of Sosky fundus dystrophy. These are patients with TIM3 mutation. You can see they have very high, thick Brooks membranes. And they're using the blood to make the RP cells or iPS RP cells. And they can see the similar high this Brooks membrane and also lipid deposit. They also correct this mutation when using CRISPR, and they can reverse this lipid deposit. But so we use these cells to do the same screenings, and they can use many nitrogen sources, including prolines. But if you look at these nitrogen sources, they're quite different from the normal iPS cells or CRISPR edited cells. They use a lot of isoleucine, leucine, valine. So all of those are branching amino acids. We also look at the culture media. In this culture media, they use a lot of prolines, like normal cells, but they use much more leucine and isoleucine and valine. Is really similar to our screenings. And this suggests these cells can use the isoleucine or leucine for their catabolism. We know the branch amino acids, they can be ketogenic. So they can generate acetyl-CoA as well as succinyl-CoAs. They can go to TCA cycle to make lipids. So one of the key enzymes is called a BC81. So we look at the BC81 expression and find in the disease cells, the expression is much higher. So this is a model we think may happen in the disease cells. They may use more branch amino acids to make more this acetyl-CoA and make more lipids, and they can cause some lipid deposit. Finally, in summary, we talk about neuroretinal metabolism as well RPE metabolism. In neuroretina, it's really glycolytic. They have Warburg effect, and they are energetic. They use about 100 million ATP per second, and they're anabolic. They involved in a lot of synthesis. And the retina, they can use aspartate and glutamate. It's quite different from other cells. And in the cone-dominant retinas, they need more pyruvate. For RPE, they are special because they are nutrient-rich. They are really reductive environment. So they have metabolism called reductive metabolism. The mitochondria are also very active. They have oxidative phosphorylation as well, and they are catabolic. So RP is really supportive. They're very catabolic, and they can use proline. I think they can also using branch amino acids, there's fatty acids in the same way to support the retina. In a healthy RPE, they are really flexible. they can use many different nutrients. But when they are diseased, they are really picky. You only restrict for specific nutrients. Instead of each of these cells, the photoreceptors or RP, they have a special metabolism, but they also complement to each other. So we see that retina RP, they're complementary and balanced. It's like a yin-yang balance in the Asian cultures. So you have to keep this balanced. In the if there's too much glycolysis in the RPE, they can potentially cause AMD. But too less glycolysis, they can cause photoreceptor degeneration. Finally, I will thank my group and thanks my collaborators, and I thank my postdoc advisor, James Hurley, and also thanks the funding agencies. I will thank you all for your attendance and attention. I'm happy to answer any questions. Thank you.
Thank you very much. We have many questions to you today, and let me just start very simply with question about dinucleotides and ADPH and ADH, whether you did profiling in photoreceptors and RPE for the dinucleotides and under different conditions like dark adapted mice or light, exposed to light.
So your question is whether we study with the light or not?
Dinucleotides and ADPH and ADH, you know, the ratio between them and how they because they're also participant.
Yeah, that's really good question. Thank you for the question. For that one, we still we have some data, but we try to trace them using different sources of these dinucleotides. But we at this moment we're still developing the method to do that. Thank you.
Okay, let's go to web and let's start with question from Tanu.
Hello. Hi, great talk. So RP is very active in mitochondrial respiration and oxidative phosphorylation, but is necessarily a glycolytic RP, is it an unhealthy state? Or as you said, it's quite flexible metabolism. It can shift between retina and RP from glycolysis to mitochondrial respiration.
Thank you for the question. I just I'll try to clarify your question. Your question is about how the flexibility of the RP and whether one of the glycolytic they can cause disease in RP, or is that a question? Is that right? Mm-hmm.
Yes.
So yeah, I think the RP that depends. They are really flexible. We culture RP and the RP in vivo is quite different. When we culture RP, they can use glucose, and the glucose actually can use lots of glucose. But if we in vivo condition, they may transport glucose to the photoreceptors.
Mm-hmm.
But if they use too much of glucose, they can, for glycolysis in the RP, they can cause problems because they can deposit to glycogens. They in vivo, they can take the glucose away for photoreceptors, then can potentially cause disease in photoreceptors. A photoreceptor can get stopped by the in the photoreceptor degeneration. I don't know whether I answer your question right.
Yes, it did. Thanks.
Thank you.
The next will be Kuitao.
Hey, Jianhai. My question is about the vivo labeled protein experiment data. I thought that Y at 5 minutes in the retina, the labeled protein is also high. Does that mean there's a transportation?
Yeah. In retina, the retina use proline because proline is amino acids, 20 amino acids for synthesis. You know, proline also a part of the outer segment as well. They use proline, but they do not catabolize. They do not break proline into pieces to make amino acid. Actually, they can take more proline than the RPE. We do not show that. Recent data we find RPE, the retina actually they make lots of proline for themselves instead of they also not only outsource proline, they can take the proline, but they do not break proline into pieces do not catabolize. It's similar for others, like leucines. We find in the RP, they can break it or catabolic, but the retina, they use it for synthesis to make proteins. They do not break them into carbons or nitrogens. I don't know whether that answer your question or not.
Yeah. Thank you.
Thank you.
Nancy,
Oh. Really great talk and high interesting work. I wondered when you stained with the SLC 6A20 antibody, it looked more intracellular than polarized to the basal membrane where you would expect it to be if it's taking proline up from the circulation. Is that true?
Nancy, yeah, thank you for your question. I think that's partially true. We try to validate right now. Yeah. Actually, when we lock out, we can see accumulated prolines in the retina. So this, I think they transport both. They can also transport prolines from the retina, but we try to validate the data. I think you are right.
Okay. Oh, maybe there's well, both catabolism and transcytosis of proline.
Yeah, I'd probably ask you more on offline for details.
Okay, thank you. Wonderful talk.
all right, let's move to Jianhai.
Hey, Jianhai. I guess I really just had two kind of basic questions from like all of your analysis. If you, and definitely from your use of different media types, if you had a recommendation for which cell culture media that people should be using to better reflect in vivo metabolism of the RPE. And if that media seems to alter RPE differentiation states.
Yeah, thank you, John. That is almost like $1 million question. That is our dream as well. We try to model the RPE in vivo, but it's quite difficult. That's we try to compare different culture media. And so far, we find the Sheldon-Miller media is better. For some other media, if you grow a little bit longer, they can also like AMD, like phenotype. But now we try to do some screenings to add different component and also reduce each component to see what are important or not. It's difficult to measure in vivo because you're not just like from the blood side, they also from interphotoreceptor matrix. Those exactly physiological condition is difficult to know. Yeah, thank you.
Daniel.
Hi, I asked the same question as John, but I guess I'll just add a little bit more to it because it seems like the issue with any kind of change, you can change medium this way, you can change it that way, you can permeabilize cells and add substrates and see that they oxidize those substrates. But I guess the big question is still, what is normal? What is ground truth? And so do you have any sort of approach for getting at that? Not just does it exhibit, say, an AMD phenotype or not, but does it look like what a mouse might do in vivo or a human might do, more importantly, in vivo? And I don't really have an answer. It just seems to be a big concern.
Yeah, Dan, thank you for your question. I think that is why we need not just doing one models, because at this moment, we do not know exactly the physiological conditions. So that is why we need to try different models, not just like in vitro models. They give our realistic advantage because reduced to simple component to analyze, but also we have to validate them in vivo to see what it's really happening in vivo or not. Yeah, thank you.
Yeah, thank you very much for your interesting talk. You showed a lot of normal consumption and production of metabolites in several different models, and I'm wondering if you've investigated or started to think about disease models for diabetes or hypoxia or ischemia, where the supply of these nutrients would be limited. Do you think that you could push the stoichiometry of normal metabolism forward to the right direction if you supplemented with these critical metabolites?
Yeah, thank you for your question. That is exactly the goal we want to do. because we want to see what is really happening in disease, whether our research can be relevant in disease or can be helpful for developing supplement or inhibit some specific pathways. But we doing this normal condition because actually we know very little about what is normal. So that is we spend too much time to understand the basic conditions. Actually, we start to doing some disease now. Yeah, thank you for your questions.
Hi, very interesting talk. I just have two questions. Most of the studies you did in the mouse, how is the human metabolism in the retina similar to mouse? And then second question I had was, have you looked into knocking out certain genes of, like, let's say, oxidative phosphorylation in RP or Mueller cells and looking into what happens to function of those, like, photoreceptors?
Thank you for the question. So the two questions. One question is human mouse. Definitely quite different. Mouse metabolism 50 times faster than humans. The heartbeat almost 300 times, much faster than us. And sugar metabolism is different. But realistically, mouse provides very good genetics. And we study human postmortem tissue. It's difficult to study the human metabolism because we can only do human models, but we find some similarities, like aspartate glutamate metabolism, both human postmortem tissues, they use it, like a pyruvate in the cone reached, they have some similarities between them, but definitely have some difference. We now analyze different metabolites, we definitely can find differences, but still, we have to accept they have differences, but we can use this model to understand the basic metabolic questions. For your second question, can we lock out this? Yeah, lockout is expensive. We are doing that now. We actually make a couple of knockouts, and the people doing that in the field as well. I'm happy to see the field is growing, and many of labs doing this. For example, in the Douglas Wallace lab, they lock out the TFAM in the RP cells. TFAM is a critical factor for the RP mitochondrial metabolism, and they can cause AMD-like phenotype. There are a couple of lockouts from other labs, like University of Michigan, from others. The lockouts key enzyme glycolysis in mitochondria in different cells, and can find a very interesting phenotype. Yeah, thank you.
Hello, I have a question. What's your opinion on possible difference in sugar metabolism between rod and cone photoreceptors? For example, I am specifically interested in what you think if cones can use glycogenolysis to produce glucose from glycogen and utilize it. Do you have any opinion on that?
Yeah, thank you for your question. So we look at because it's difficult to study this, difficult to isolate cone, and we can study using two methods. You look at the gene expression between the cone photoreceptor and the rod photoreceptors. Because no single cell database is easily available, you can see the glucose metabolism definitely really high in the cone photoreceptors. And they have expressed the glycogen phosphorylase is a key enzyme for glycogen degradation, mostly in the cones, not too much in the rods. So the glucose metabolism is quite different in cone and rods. The cone can utilize glucose well than the rods. And also another way is like what we did. We can just simply incubate that. And now we try some way to remove that. We remove the cones to see. And I'll compare those tissue with or without cones to compare their metabolism. Yeah, thank you.
Hey, Jianhai, this is Vladimir. Very interesting work. Thank you for presenting to us. I was wondering about the difference between the diurnal and nocturnal species, but Golap already asked you that. So I'll ask, what is known, and can you tell us a little bit about the transport of nutrients from the RP to the retina? How is that organized?
Thank you for your question. Yeah, the transporters from the RP to the photoreceptors, they are really, really active. There's if you look at the most abundant genes in the photoreceptors, the top genes are like rhodopsin or those phototransduction genes. But if you look at the top genes in the RP, they are transporters. We seriously look at transporters as about the total about 400 transporters. But in the RP, at least 200, 300 is really, really abundant. They can transport some glucose, like Nancy did a very nice work on glucose transporters, lactate transporters. They also transport GABA, taurine, or thyroids. So transporters definitely very important in the RP and is not well appreciated. I hope that more people can study that to understand this transporter in the RP.
And if I can follow up, so are those nutrients just released in the extracellular space and then taken up by photoreceptors, or are there vehicle vesicles that transport them between cell types?
Oh yeah, that's a good question. Those one I do not know. They can be actual vesicles, but I don't. There's too much research on that. But they can be two ways because RP is polarized. They can be apical side and basal side. That's as Nancy asked the question. They can be different. For example, the MCT, the lactate transporters. Some of the transporters go to one side, another can be another side. Then you would know they're coming because it's two ways. Some coming from the blood side, some coming from the photoreceptor side. It's like a two-way or one way. Some of them can be vesicles, but a vesicle I do not know too much about that. But in other cells, like endothelial cells, seems like there are very active research on that. May be important in the RP as well, but we do not know. Definitely, we can see some vesicles when we do EM for some culture media. We still do not understand those. Thank you.
Okay, thank you.
Hi, nice talk. So I was wondering, more close to the basics of biology, I find it interesting that you're looking at the nitrogen isotope similar to how weighted carbon dioxide is used to trace the general metabolism. And I find it very interesting that arginine was not used as a nitrogen-rich amino acid in the RPE in retina. Why do you think that the metabolism used glutamate and aspartate instead of real nitrogen-rich side chain such as arginine?
Yeah, that's a good question. We do not know for that as well. That's why we do these screenings, and we thought they can use more. But that is aspartate, glutamate. I think they have special metabolism because they can help. We did not show here. If they use aspartate and glutamate, they can assist the Warburg effect because aspartate can convert to something. They can help the sugar transport it and help the glycolysis. So as the arginine, we do not know why arginine do not use it too much. But the problem for the arginine, if they use or degradations, for example, glutamines, they are nitrogen rich. Asparagine, they're nitrogen rich. The nitrogen rich, if they use this too much, they can release ammonia. And ammonia can be potentially toxic to the neurons. And we think glutamine sometimes increase because they can dance to take this nitrogen to make a not toxic glutamine or arginines, then export it to detoxify. This is what we think. Thank you.
Hello. Hello, thank you for the talk. Kind of building on that question, when the mice were injected with the proline containing the nitrogen isotope, was there a preference for generation of a certain amino acid when you collected the time points during the course of the study?
Yeah, thank you. So the question is, any preference for some amino acids from the nitrogen proline at different time, right? Yes. So we find the proline definitely can easily convert to glutamines and glutamates. They can also generate about, we find in vitro about 13 amino acids. In the mice, about 8 amino acids they can make. And the glutamates, glutamine, and aspartate, they are make more from this C13 proline injections, and also anilines. Thank you.
Hi, Jianhai, this is Dorota. Nice to see you again. And great talk. I have a question, and you probably know what I want to ask. Does metabolism of these amino acid change in aging?
So amino acid, yeah, nice to see you virtually again. So amino acids changes in aging. Yeah, that's a great question. Actually, we try to understand that. By the proteomics, we find they are different, and we published a paper before. In the aged mice compared to the young mice, the amino acids is quite different, especially the methylated amino acids, because one of the hallmark for aging is epigenetic modification. They have lower methylation or hypomethylations. But in the aged retinas or RPs, the methylated metabolites, especially methylated amino acids like trimethyl lysine, methylhistidine, or these methylated amino acids much higher in the aged mice. Thank you. I think probably this can take away this messy group and make the less messy group to modify the DNAs. So they may be one of the mechanisms for the agings. Thank you.
Hi, Dr. Du. This is Emily, also from Dorota's lab, who, of course, asked my question about aging. I have another question about proline utilization in the RPE. Have you or others looked at maybe iPSCs derived from patients with AMD or AMD tissue to look at this proline utilization in the RPE of AMD patients?
Thank you, Emily. We are looking at that now because some of the other, we collaborate with a couple of groups, they're doing AMD. We are actually looking at that now. But I don't think the proline is the major one from the data now, but we cannot show the data, but we probably can know that soon. I hope that a proline can be major changes, but unfortunately, so far, I do not see proline as a major changes in these AMD cells. Yeah. Thank you.
Hi, Dr. Du. Great talk. Thank you. I now have a question about the pyruvate metabolism in cone cells. And can you comment on why cone cells need more pyruvate for metabolism? It is because of the energy demanding, or like because it need more of it to synthesis fatty acid? And I'm asking this because I think there's more views like launching polyunsaturated fatty acid in rod cells more than cone cells. And the second question is the consumption of pyruvate in light or in dark? Because I think the energy requirement for cone cells is different in light and dark. Thank you.
Thank you for your question. It seems like you know the question, you know the answer already. For the pyruvate, I'm not sure. We try to understand this as well. We think a pyruvate can be direct substrate for because the glucose is still needed to glycolysis. If you look at the cone cells, compare with the mitochondria, I think Dr. Hurley showed very nicely before. In each rod photoreceptors, they are about like I remember it's 12 or 13 mitochondria. But in the cone cells, almost all mitochondria, it's like hundreds of mitochondria. So the mitochondrial metabolism or oxyphosphorylation can be pretty active in the cone cells. And the pyruvate can be very important like fuel or substrate. They can directly go into cone cells. And if you look at the gene expression like pyruvate dehydrogenase, pyruvate carboxylase, they are pretty higher in the cone cells. That is my speculation. So we try to lock out these genes in the cone cells to see how important they are. And the second question in the light dark, I think, yeah, we did not do that. I did only doing this in Jim's lab. At that time, actually, I do not like dark too much. So we did not do the dark experiment yet. Yeah. Maybe you can do it or someone can do it. Yeah. Thank you.
Thank you. Hi, Dr. Du. Thank you for your talk. I was wondering in your study where you blocked the transporter, how it affected the rate of glycolysis in the retina. And I remember reading that like glycolysis is an inhibitor for fatty acid catabolism. And like, I was wondering if you found any evidence of higher ketone bodies in the retina when you blocked blocked the transporter.
Yeah, thank you. Thank you for your good question. So we still do not know yet. We expect may have the changes for glucose metabolism, as you said, because they are balanced between the glucose metabolism and the fatty acid metabolism, because they have a cycle, they are inhibit each other. But we are still expanding lines because of COVID. We have the line already, but then we generate line and lose it, expand it. Now we have not doing that. Hopefully in the summer, now we got more mice. We probably know this answer soon. Yeah, thank you.
Well, we wanted to thank you for a wonderful presentation, and I hope we learn a lot. So until next week, bye.
Thank you all. Thank you for your attention.